Hello everyone. Let's study principle of inheritance and variation, subtopic codominance. What is this codominance? Codominance, that is also exception for the law of dominance. Already we studied the incomplete dominance and this is the second one, codominance. Okay. The codominance is a genetic pattern where both alleles for a gene are fully and equally expressed in a heterozygous individual, meaning neither allele masks the other. Example, AB blood grouping in human beings. So here according to the law of dominance. So in the heterozygous condition, for example, capital T, small t is there means, so only one is expressed, right? So here capital T is expressed, so the plants will develop into tall plants. So here small t is recessive, right? So it is not expressed. But in the codominance, both of these characters are expressed. Means, so there is no dominant or the recessive. Both the alleles are equally expressed in the heterozygous individual. So for example, AB blood group is example for the codominance. So let's study the how this AB blood group is example for the codominance. Okay, here this blood group in human beings that is controlled by a gene that is called as I gene. Okay, so blood grouping in the human beings, you know that in human beings B, AB and O blood group is there, right? So these are the four important blood groups that is found in human beings. So this blood group is controlled by a gene that is called as the I gene. Okay, so this I gene that has the three different form. So height of the plant that is having the two different genes, capital T or small t, right? So these are the two different alleles of which is responsible for the height of the plant. Same for the blood group. For the blood group, so I gene is controlled by this I gene which control the blood group type. So it has three different types, that is three different alleles, that is IA allele, IB allele and the small I allele. Okay, so this I gene that is responsible for the plasma membrane of the red blood cells has the sugar polymers that protrude from its surface, and that kind of sugar is controlled by the gene. Okay, so the red blood cells, just imagine this is the red blood cell. So this red blood cell that has some sugar polymers on its surface. For example, these are the sugar polymers that is present on the red blood cells. Okay, so that is controlled by the I gene. So if IA gene is there, so here the sugar type will be IA type sugars will be produced. IB means IB type of the sugars will be produced. Okay, so the I gene that control the which produces the sugar polymers on the surface of the red blood cells. The alleles IA and IB produce slightly different forms of the sugar, while allele small I does not produce any sugar. Okay, just imagine. So this is the red blood cell. So there are some type of the sugar polymers that is by the IA. IB means that is slightly different. Just imagine IB is like this, and small I gene is there means that do not produce any sugar polymers on the red blood cells. Based on this sugar polymers, the blood grouping is done. Okay, because of human are diploid organism, each person possess any two of 3I gene or 3I alleles. You know that human beings are the diploid. So there are three types of the alleles, IA, IB and small I. Okay. So here the human beings are diploid, so they possess any two of these three alleles. It may be IA, or it may be IB, it may be IA small I, or IB small I, or IAIB, or small I. So in the human beings, so they are the diploid organisms. They possess any two of this allele, any two. It may be IA, IB, or the same allele or the different allele. So here IA dominates over small I. Okay, so if IA small I is there, so IA is expressed. The blood group type will be IA. So IB also dominate over the small I. So if IB and small I gene is there, then the blood group will be B type, because IB gene is there. So if small I is there, means so that is there is no sugar polymer, so that blood group type is called as O blood group. So in case IA is there, IB also is there. So the blood group type is AB, because here IA also expressed and IB also expressed. So the blood group type is AB. Okay, I'll clear it once again. In case of IA is there, the blood group will be A blood group. IB IB is there, the blood group will be B blood group. Okay. So here IA small I is there, then the blood group is A blood group, because this capital IA that dominates over the small I, and IB small I is there, then that is B blood group. Okay. So in case small I is there, that is called as O blood group. So the special case IAIB is there, then the blood group is AB. Here IA is not dominant over IB, or IB is not dominant over IA. So here both the alleles are expressed. So the blood group type is AB blood group type. So this is an example for this AB blood group is an example for the codominance, exception for the law of dominance. See here. Here you can see IA and IB are completely dominant over small I. So IA and I are present, only IA is expressed. So small I is not expressed, and when I is present, then IB expresses. So but when IB are present together, they both express their own type of sugars. So the blood group type is AB. Okay, so that contain both A type of sugars and the B type of sugars also. So that is example for the codominance. And one more concept, multiple alleles. So what is this multiple alleles? For example, AB blood grouping. So here the single character that is controlled by more than two alleles, that is called as the multiple alleles. For example, blood group of the human beings. Okay, blood group of human beings that is controlled by single character, that is controlled by more than two alleles, that is IA, IB and small I, three alleles, right? So that is example for the multiple allelism, or it is also called as multiple alleles. There are more than two alleles governing the same character. So these all are controlling the same character, that is the blood group type, right? So this is example for the multiple alleles. So here you can see this table. If the parents having the IA and parent 2 also having IA, then the genotype will be IA. When IA condition is there, the blood group will be A blood group. And similarly, you can see the B blood group and the O blood group condition and also AB blood group. Okay. So this is example for the multiple alleles. And one more example is given here. A single gene product may produce more than one effect. Here, starch synthesis in pea seed is controlled by one gene. It has two alleles B and B. Okay. So the starch synthesis in the pea seeds, that is controlled by one gene. It has two alleles, that is B and B alleles are there. Starch is synthesized effectively by BB homozygous condition. The starch synthesis is effectively conducted when BB condition, homozygous is condition, and large starch grains are produced in the pea seed. The BB small b condition, homozygous, has less efficiency, hence produces the smaller grains. So BB produces the smaller grains, capital B that produces the large starch grains. After maturation of the seeds, BB round and the BB seeds are wrinkled. The heterozygotes, when you cross this, the heterozygous condition contain BB and the small b. Produce round seed, and so the B seems to be dominant, but the starch grains produced are intermediate size. So here the size of the seed, that is round. Okay, so here BB means that is round, small b means wrinkle. Okay, so capital B means that is producing the round seeds and the large starch grains, and here small b that is wrinkle and producing the small starch grains. So when it is crossed, the F1 or the heterozygote that contain the capital B small b, the size of the seed, that is round only, so that is round only, but the starch synthesis, that is intermediate, so that should be also large. No, large starch grain should be produced, but it also produces the small starch, intermediate starch grains. So here, if the starch grain size is considered as the phenotype, then from this angle the alleles show the incomplete dominance. So if you see the synthesis of the starch as a character, then so this alleles also show the incomplete dominance, because in the F1 generation they produce the intermediate size of the starch in the grain, right? So that is also example for the incomplete dominance. Thank you.